we will pass the mic over to Dunling. Over to you.
Good morning. Yes, we only have 6 meetings a year. There's so much information we need to pass on to producers. So we decided to squeeze a little bit information in one presentation. This is a typical one. As Marna said, it's cover crops, on-farm trials, and adopt information. So I try to merge them together and give a better name. So it says on-farm trials, on cover crop, and adopt. Let's see how it goes. Some of us would ask why we, as a producer, why I should do research, because there are scientists who does this for their career. And I want to give you a little bit explanation why you need to do that on farms. First of all, the research done by scientists with a small plots experiment is for clarity of science. So they always choose to use a flat and uniform land to avoid any variations. Number two, they rarely consider the cost to grow a crop on their experiment. And more importantly, number three is a significant difference in scientific term may not may not be important to actual crop production from producers' perspective in many cases. And a second, this on-farm trial would provide a chance for you to make the science work with your skills and with your own farm equipment on your own land. In other words, to check that if the science developed by those scientists would work on your situation. And number three, this is an excellent opportunity to attest the new approach works on your farm. More importantly, this is an experiment that you will decide if the new approach that you plan to adopt on your farm will produce a real profit for you. And first, let's look at the cover crops. Before we're talking about cover crops, I would like to spend a minute to review the original objective, why we grow cover crops. In many cases, we forgot about the original objective. Of course, the cover crop would be able to prevent soil erosion from the wind or spring runoff water. But in addition to cover the soil, to prevent soil from erosion by wind and runoff, I think there are two major objectives. The first is the cover crop are an investment for future crop production and building the system for your crop production. And a second is to provide the nitrogen for your following crop for high yield and quality. And the second objective is to produce a large amount of biomass to build soil health by increasing soil organic matter and to improve soil biodiversity. Did I move that too? I think I have a little bit issue with my computer here. Okay, here we go. Cover crop, grow multi-species cover crop. Cover crop is an independent crop that will take a whole season to grow. but it is not actually an independent crop that will bring you a financial profit. I think when we consider cover crops, we should consider cover crop as a part of your crop rotation. So treat the cover crop as an investment for future crop production and building your system resiliency. And a successful cover crop can only be reflected by how well it serves the cash crop production in the next couple of seasons. Numerous studies have shown that multi-species cover crop outperform the single species. Because multiple species cover crop increase the efficiency and efficiency on everything, like produce biomass or fix nitrogen and booming the soil microbial activities. And multiple species cover crop should include all the crop categories, including legumes, fibrous roots, and brassica crops. And it should also include shallow-rooted and deep-rooted species, as well as the drought and flood tolerant varieties. So that would give you extra guarantees that the cover crop would perform. This slide reviews some of the key benefits of multi-species cover crop versus, like a single species cover crop. The first of all is the trellis effect to support climbing crops. For example, pea and wheat, like wheat would be able to support the pea to grow upright and make it grow better, not really shed it on the floor. So pea would be able to produce more biomass and fix more nitrogen. And the second is that multi-species often result in a reduced pressure from disease and pests. And third one, also increase land equivalency ratios. That means produce more than the single, the land use, the LER is normally more than 1 when you grow multiple species. That means increase your land use efficiency. And when different species crop grow together, the nitrogen use efficiency can be improved through the nitrogen transfer under the ground. From the legume crops to the non-legume. And some preliminary research have also indicated that it might be able to mobilize the unavailable phosphorus in your soil as well. Of course, we need to focus our attention to legume crops when we grow cover crops. To ensure the nitrogen fixation, we normally recommend 3 varieties of legumes to be included in the multi-species cover crop. And these 3 legumes should be different in many aspects, such as growing features above ground and underground. Some have a stem they could stand along, others would climb, or they have deep roots or shallow roots. Some are drought tolerant, others would won't die in flooded water for several days. Research work done at the University of Manitoba suggests that the total legume biomass should be greater than 50% of total biomass at the time of termination for maximum nitrogen fixation efficiencies. So this kind of ratio can be controlled by mixing the seeds. I mean the seed ratios. You need to put more than 50% of the legume seeds in your cover crop mix. And another important aspect is to measure or estimate the total amount of nitrogen fixed in your cover crop seasons. Because the nitrogen fixed in your crop would be able to decide what kind of crop rotation you would use. After growing cover crops, the total amount of nitrogen brought into your soil is estimated. You can plant your cash crop for next 2 to 3 years based on your cash crop nitrogen needs. Here's an example. If we grow a cover crop in first year, they produce 5,000 dry biomass, and with nitrogen content 2%, then you actually fixed 100 pounds of nitrogen in that cover crop year. And the second year, you could grow a wheat. And if you are aiming for 30 bushels yield and 14% protein content, that means every bushel of wheat would export 1.9 pounds of nitrogen. So altogether, second year for this wheat crop, you would lose 57 pounds of nitrogen from your soil to produce this wheat, and then you still have like 43 pounds of nitrogen left in your soil for your third year crop. So if you decide to grow a flax, which we often say they don't need a whole lot of nitrogen. As a matter of fact, the nitrogen content per bushel of flax is slightly higher than the wheat. We often think it's not really a nitrogen required crop because the yield is lower. So if we are receiving 14 bushels per acre yield with 2.12 pounds nitrogen per bushel, you actually put 30 pounds of nitrogen into this flax. So after 2 years of cash crop production, your soil still have 13 pounds of nitrogen left. What are you going to do with this 13 pounds of nitrogen? If you try to grow another non-legume crop, it may not support a great yield by this nitrogen. And well, if you do grow, let's say, oats in your third cash crop year, the crop may yield higher than those nitrogen available in the soil because the crop would be able to dig into the nitrogen in your soil organic matter. So actually during the growing season, soil organic mineralized and provide nitrogen to your oats growth. But the cost is you lose your soil organic matter. So which is not what we really want to do. For this 13 pounds of nitrogen left in the soil, you could do several things. One is to go back to your cover crops, start the cycle again, or you could leave this 13 pounds of nitrogen, which would help to grow your cover crops, because cover crop need nitrogen as well to grow, even they are legumes in there. Some of the species in the mix, which doesn't fix nitrogen, need the nitrogen in the soil to take off. Or you could grow an intercropping, and this 13 pounds of nitrogen would only use to support non-legumes and share some of the nitrogen fixed by the legumes in that season. And then you probably could have a pretty good crop. And always think about, these things before you go for the crop, for the cash crop. And now let's move to on-farm trials. I summarize it into 6 steps. Now let's go step by step. Step 1 is to ask a specific question that your on-farm trial will provide answers. The question should be simple and only one. For example, does a cover crop with multi-species provide more benefit than single species cover crop? And then you could start thinking about on-farm trials around this question, and allow maximum 3 treatments. Too many treatments would make it very difficult. And if you are a skillful person, did this before, I would suggest to use 3 treatments. Otherwise, 2 would be more suitable for most people. More treatment will may cause interaction and make the result difficult to explain. So leave other kind of questions for another experiment in future years. And it'll probably a good idea before you start. You could talk to the farmers and partner with several others, and which would help you to, build your research ideas and ask a better question. And if you want, you should, if you want to, you could also include a professional consultant. When you have some kind of questions, you could, go to them, get answers. And step 2 is to keep experiment simple. Again, I would say allow maximum of 3 treatment. Something like, if you do a cover crop mixtures, you could do a mix of 3, 8, or 16 different species. So those are the treatment. And plus, you would always have, if you do multiple species, and you probably use a monospecies as a control or as a comparison, include what the second part would include what you use to grow as one treatment for comparison purposes. That is called experiment control. And normally I would suggest seed 3 strips of each treatment across the field containing all kind landscape positions. And this 3 treatment will be repeat. The repeat doesn't have to be in the same field. You could actually, design this and do 3 different fields. And those fields is where that you are going to grow your cover crops. Step number 3, choose a site and lay out the treatment. And the best location is the field where this practice will take place. But avoid unusual spots, such as old farmyard, old yard location, or barns. And something like, abnormal situation may be there. So you want to avoid that. And run strip cross the slope positions, not parallel to the elevation, because in that case, you would cover all landscape positions and see how it performs on different kind of landscape, on nose, on mid slope, and the lower slope. Again, the repeat can be in different field, and randomize the repeats, not always put a control besides the road or ditch. Step number 4 is define parameters to measure and decide when and how to measure them. And for the cover crops. Biomass production is one parameter, and the nitrogen fixation is another. So, you could measure those by collecting all the plant in unit area and say, a square meter at several locations in each strip, which cover all the landscapes, positions, like, choose those sites on different locations. More sites will give you a better results. And remember, randomly do that, not to choose too many sites on particular landscape, and evenly distribute those. The second parameter you might want to see, you want to see the biological nitrogen fixation of cover crop. And this can be done as well by this kind of a sampling. And you would collect those biomass and measure how many pounds of the biomass produced per acre basis, and then times the nitrogen content, then you would be able to get the nitrogen fixed by your cover crop. And nitrogen content can be measured at the labs. Many labs will do this kind of plant material analysis. It's a pretty simple test, take only a few minutes in the lab. You should get the results pretty quickly. And I would suggest you would take for soil sampling to analyze the soil organic matter, nitrogen, phosphorus, and other soil parameters, such as, if you're interested, you might want to see the soil microbial community structure that would show your microbial diversity in your soils, under different kind of mixture. Number 5 is stick to your experiment design and keep records. The important part is following through your design and leave your ideas for next project. I'm saying this because a lot of times when we do experiment, when we see something happened, we got a new idea, and we want to insert this idea into the existing experiment, and which would mix up all things, and eventually you don't know which is which and what is that for. Follow through your design and leave your ideas for next project. If you do feel like this is something that is interest to you, do it next time. And then you would ask a slightly different question. And have a notebook with you all the time. Write down whatever you did, when you did it, and where you did, and what you saw. And many unexpected results often explained by the conditions during the growing season. And when you look at your notes, you probably have this kind of aha moment. And support this record with the photos, and you have a cell phone, and which would you can take pictures to support all those kind of things you did in the field. And at a later time, it will be easy for you to review those kind of data. And the number 6, the final step, is to analyze the data you have collected and conclude the trial. When you put all the information together and compare the results with your controls, you would see which one is better. If you need help with statistical analysis, ask a professional. And by this time, you would be able to conclude if the trial is successful. I mean, that if the idea you test work on your farm. So even it didn't work as what you planned, it is still a successful trial, because that thing doesn't work on your farm, to avoid a big financial loss. So I would congratulate you that you have completed your on-farm experiment. By this time, you would answer the question you asked at the beginning of your experiment. And now let's move to agriculture demonstration of practices and technology program. The program provides funding to help agri-arm sites and producers groups demonstrate and evaluate new agricultural practices and technologies at the local level. Sasa Organics is eligible for that. And the results of successful trial can then be adopted by farmer, in your whole region. So, and eligibility and eligible recipient include incorporated agri-arm sites and Saskatchewan-based producers group, including Sasa Organics. But as individual producers, we are not eligible to apply under this program. But you can access the funding through your producers group. For example, if you want to do a test, test idea on your farm, you could ask Sasa Organics to make an application on your behalf, and then you would be able to use the funds for the experiment. So you can manage and conduct adopt project on your farm and receive funds through the project awarded to your producers association. Generally, the funding is awarded for projects with a 1-year duration. Exceptions will be considered for requiring more than 1 year to complete. In many cases, organic production system require an additional year to see how the results show, how the practices show the results. So the 2 years is, we have received 2 years of funding for some of the organic related on-farm experiments. The funding is awarded on a competitive basis. Each year we receive about 150 to 200 projects, but we would select some of those projects that is useful not only to your own farm but also your neighbors. Like once that kind of trial is done, the whole region would be able to benefit. That's the thing we are looking for. And each project is eligible to get funding up to $10,000 per site per project, to a maximum of $75,000 per year for an organization. So it is a good idea to, you could partner with several farmers from different regions in the province and conduct the similar kind of demonstration or trials. And then that will be, you got multiple sites, it become more representative, and you got more funding, and then the results will be, be able to demonstrate to different people, to many people in the province. Eligible expenses, including all direct project costs like material, seeds, or something, but not equipment. Administration cost up to 5%, and field day cost to a maximum of $1,000. So you could use $1,000 for your field day demonstration. Ineligible cost, any capital purchase over $300, because the ministry doesn't want to look after the equipment you purchase for the project after the project is done. So if you purchase capital, remember, keep the cost below $300, and then it will be like a consumable. And honorarium for staff or producers member is not eligible as well. The application deadline, there are two applications accepted for a year. For projects starting in fall is June 7th. So some of the project, if you need to make a preparation for the spring treatment, for spring experiment, and you could apply in July 7 around. And July 7 normally have less applications, and it's easier to get funding. That's well, did I say that? Most of project application will come in November. We have a huge number of application in November, which will be for the project for the next spring. Yeah, that's my presentation for today. And to summarize, and I would say a success of cover crop is evaluated by how well this cover crop support profitable crop production next 2 to 3 years. And I would encourage everybody use multiple species cover crops for maximum biomass production. And I would also suggest include 3 different legumes in multi-species mix to ensure efficient nitrogen fixation and reduce the risk of climate conditions or whatever. And plan your crop rotation based on the soil nutrient balance. A surplus, well, basically based on your soil nitrogen content or soil nitrogen that was fixed by your cover crop. Remember to leave a surplus small amount of nitrogen in soil for building your soil health, not to dig into your soil organic matter, because when the microbial work on the crop residue to turn them into soil organic matter, the microbial would need nitrogen to do the work. Incorporate a legume crop to boost nitrogen supply anytime during the crop rotation period. Design and follow through your simple field trial to answer a specific question that you asked for the field trial. Analyze experiment data to examine if your idea will work on your farm. If it's not, don't do it. Well, you could call me if you are interested in conducting an adopter project on your farm, and I would give you more ideas and see. I probably could help you to draft the proposal as well. Yeah, either you could contact Marla, Deb, or myself, and we will work together and get this going. Yeah, that's my presentation. Thank you.
Perfect. Thank you, Dunling. So